Okay, so we're at the top of the hour. Very pleased today to be showcasing a topic of human islets, proteomic predictors of individualized nutrient-specific insulin secretion in health and disease. We have two scientists with us today coming from British Columbia. At the University of British Columbia, Jelena Kolic completed her PhD in 2014 with Dr. Patrick McDonald at the University of Alberta. Sorry, just letting people in. Her thesis focused on examining the role of the PI3K signaling pathway in insulin secretion in human islets from donors with and without type 2 diabetes. The main finding of her work showed that the inhibition of this signaling increased insulin secretion from normoglycemic donors, but failed to do so in donors with type 2 diabetes, showing that this pathway is perturbed in disease state. In 2016, she joined the lab of Jim Johnson as a postdoctoral fellow, and her current research interests explore heterogeneity in insulin secretion responses to nutrient stimuli and other fuels. Dr. Kolic's currently on the job market. She's looking forward to starting her own program and research, and she's particularly interested in the interplay between genetic and environmental factors that mediate the regulation of insulin secretion and discerning why some islets are more susceptible to dysfunction. We also have with us Dr. Howard Sen. He's received his PhD in Genome Science and Technology program at UBC as well. And currently he's a postdoctoral fellow in Dr. Jim Johnson's lab at the Life Science Institute, UBC. With his training in molecular cell biology, physiology, and bioinformatics, Dr. Sen's research dives into insulin action and resistance, employing systemic multi-omics analyses, and his work primarily focuses on the peripheral tissues in both primary and stem cell islets. Welcome to both of you. Thank you so much for joining us from a beautiful Vancouver. And we are very excited to hear both of your presentations.
Thank you, Monica, and thank you for the really kind introduction. So yeah, I'm Jelena. I'm a research associate in the Life Sciences Institute at UBC, and I work with Dr. Jim Johnson in Vancouver today. I don't know if you can see, it's not so nice, but we're getting a little bit of rain, but hopefully we'll see some sun later today. So I would like to acknowledge that we're actually virtually gathered here today on the traditional ancestral and unceded territories of the Musqueam people. So I both live and work on the UBC campus, and the research being presented today was conducted at UBC as well. So primarily today, I'm going to be giving a brief overview and sharing with you some of the highlights of a recent paper that was published in Cell Metabolism. I'm going to be focusing on islet functional data, so human islet function. And what I mean by function in this context is insulin release in response to 3 model macronutrients, so glucose, amino acids, and fatty acids, as well as looking at the proteome of each donor that we looked at and how the proteome could be used to better understand islet function. And I believe Howard will take over just shortly halfway, talking a little bit about the transcriptome and how the transcriptome and the proteome may or may not correlate with each other.
Great.
I'm also going to take a few minutes just to talk about the Human Islet portal, the web portal, how our data was integrated into the portal, and how you could use the portal to sort of dive deeper into the data set that's found in the Cell Metabolism paper. Okay, so just a little bit of background to put this research into context for you and to sort of talk about, why we decided to do this study. So we've known for quite some time now that our bodies respond differently to food. And so what I mean by that, at least in this context, is that postprandial blood glucose levels are going to be different between different individuals, even though they might be ingesting the exact same meal. So for example, if you were to give, 2 different individuals an apple of the same size, same weight, same type, their blood glucose levels would not be exactly the same. And this was shown in 2015 by Zevi et al. On a much larger scale, where they looked at 800 individuals, normal glycemic, so they did not have diabetes, but they searched for individuals of a wide range of ages, HbA1cs, and BMIs. And what they found that in response to a standardized meal, their postprandial glucose levels. So what I mean by that is the level at which their blood glucose levels rise to following this meal were very different, right? So we had a large heterogeneity in postprandial glucose responses to a standardized meal. And you can see this in more detail here. So for example, these 800 participants, they were given, pure glucose, bread or bread plus butter. And what we're looking at here is the postprandial glycemic response. Yellow indicates a higher response, blue indicates a lower response. As you can see here, most of us would expect that pure glucose would cause, quite a large spike in blood glucose levels, and it does in the majority of the individuals. However, there's also a lot of blues here, right? Suggesting that in some individuals, glucose did not cause as large of a spike as we would expect. And this is one of my favorite examples to show. I use this quite frequently. But here we're looking at two different individuals from the study and their postprandial blood glucose in response to either a banana, shown in yellow, or cookies, shown in blue. And so typically, most of us would expect the banana to have, a lower glycemic index and cause a lower postprandial blood glucose increase. And you see that here with this participant. But in this participant up top on 445, you see the opposite effect, whereby the banana caused a higher glucose spike than the cookies, right? So again, reiterating that heterogeneity or something known as inter-individual variability in blood glucose levels. And this also seems to be true for individuals living with type 2 diabetes. So this was a study done in 2020, a much smaller study. They only looked at 23 individuals. But again, we're looking at blood glucose rise in response to fructose, glucose, bread, or bread plus butter. Each one of these traces is one individual. And again, you see that, that variation or variability between the different individuals. And again, here's sort of a more extreme example. We're looking at glucose in green versus bread plus butter in blue. Again, two different participants. We're looking at the glucose rise. Most of us would expect pure glucose to cause a higher spike in glucose levels, and it does in participant 2, but not in participant 1. Again, reiterating that inter-individual variability. And I don't have the data to show you here today, but some recent studies are showing that this also is likely true for individuals living with type 1 diabetes as well, where you see that high inter-individual variability. So this is where our study comes in, and we wanted to find out if insulin secretion, which of course insulin is released in response to rising blood glucose levels, if insulin secretion in response to specific macronutrients, so not just glucose, but other nutrients like amino acids and fatty acids, if that's individualized as well, whether we're going to see that heterogeneity that we see in postprandial blood glucose responses. So how do we measure this? So we went directly to the source of insulin, and that's of course the pancreatic beta cell, which is found within the pancreatic islets of Langerhans within the pancreas. We examined islets from 140 human islet donors and counting. We had both male and female donors of a wide range of ages. The islets were all isolated in Alberta at the Alberta Diabetes Institute. And they were shipped to us overnight after one of the first things we did was islet functional experiments. And again, when I'm talking about function in this context, I'm talking about insulin release in response to our different nutrients. So glucose as our carbohydrate, we use leucine as our amino acid, and oleic and palmitate as our fatty acid stimuli. So long story short, the same inter-individual variability you see in postprandial glucose responses in response to different meals, we also see at the level of the islet when it comes to insulin release. So what you're looking at here is insulin secretion on the y-axis over a period of time in response to different concentrations of glucose, amino acids, so leucine alone, or leucine together with glucose, or oleic and palmitate as our fatty acids alone or together with glucose. And so each one of these squiggly lines, as you would say, represents one human islet donor. We looked at 140 preps and counting. In the teal, what you see is the average response from the donors without diabetes. And in the salmon is the average response of the donors with diabetes. And these floating dot plots indicate the calculated areas under the curve for the corresponding section. So what I hope you can appreciate from this is they're not just squiggly lines. Each one of those lines is one human islet donor. And what you can see is that high inter-individual variability. And this becomes even more striking when you try and do this experiment on mouse islets, which most of us in most labs would do. And the interesting thing is most of us would probably focus on mouse islets from, one sex, either male or female, of a particular age. And most of us would use an inbred mouse model, which we did in this case. So these are islets from C57 black 6 mice. But we did try and search for this heterogeneity a little bit by using islets of both sexes and islets of mice from different ages. So as young as 7 weeks and as old as 90 weeks. And what you can see here, or I hope you can see, is that variability that we see in the human islets is really absent in the mouse islets, right? We can't find this. And so if you're only studying mouse islets, you're probably not going to see this variability that we see in the human islets, or at least not at this scale. So then the question we wanted to ask was whether this variability is biological. So can we use proteomics or RNA-seq data to better understand this variability in insulin secretion? And so for the talk this morning, I'm primarily going to focus on the proteome, and I believe Howard will talk a little bit about the transcriptome later on. So again, we received islets from Alberta, from Edmonton at the ADI Islet Corps, 140 donors we've looked at and counting. They were shipped to us overnight, after which we did functional experiments. So we looked at insulin secretion in response to the different nutrients. But we also, for every islet donor, we also sent off for proteomics analysis. So we had information about protein abundances with every islet donor, as well as we collaborated with Dr. Anna Gloin, where we were able to get gene expression data for most of these donors. Now, as you can imagine, we now have the transcriptome, the proteome, and functional data. And now we can do some really cool analysis whereby we can try and understand whether, islet function in response to a specific nutrient is also correlated with, the abundance of a specific protein or the expression of a specific transcript. And long story short, yes, that is the case. So we found, hundreds of proteins that were either positively shown in red or negatively shown in blue, associated with different functional or donor metadata parameters. So again, we also had information about age of the donor, sex of the donor, glycemic control, and so on. And a number of these proteins sort of served as a sanity check because we knew that they're involved in insulin release. So for example, phosphofructokinase, shown here, has been pretty well studied and is known to influence glucose-stimulated insulin secretion. But again, there's hundreds of proteins here, and a lot of them, a vast majority of them haven't really been explored and haven't been looked at in the context of insulin release, type 2 diabetes, beta cell function, and so on. And for somebody who's looking to start a lab soon, hopefully, this is sort of a very vast abundance of data set that we could follow up on and try and figure out signaling and what happens in diabetes and how these patterns or proteins are perturbed.
It even seems, to interrupt quickly, Jelena, it even seems like a great place to play in terms of mixed meal responses. Yeah. And you have like a whole palette here. You could really do so many experiments. It's really exciting.
For sure. Yeah, that's something else we didn't do as much. We mixed glucose a little bit with the amino acid or the fatty acid, but it'd be really interesting to do like a in vitro mixed meal test, which isn't done routinely in the lab for sure. So
yeah.
Can you explain the experiment? I mean, is the idea that you give these different treatments for 20, 30 minutes or something, and then lyse tissue and do proteomics after a short-term
No.
exposure?
Good question. No, so the proteomics was actually done on untreated islets. So they were not islets from the same donor, but before they went through the perifusion protocol. So I do apologize for not making that clear. So we had the same with the RNA-seq. So that was done on untreated islets. So these islets did not see, the glucose, the amino acids, or the fatty acids. So this is a perifusion profile. So we treat the islets with low glucose initially, so 3 millimolar glucose to get a baseline. Then they are treated either with high glucose, leucine alone, or palmitate alone at the basal low glucose levels for 40 minutes. And then you bring them back down to 3. And then we treated them with a medium dose of glucose, either alone or with leucine or with oleate and palmitate, brought them back down to 3, and then finally treated with KCl. So those 2 experiments were done, these were done at the same time, on 6 different, 6 different lanes of our perifusion. So same islets from the same donor, I should say. And then we had a separate batch of islets that we flash froze for proteomics on the same day.
And can you just say a word about what the conditions are for the islets that are going to get the proteomics?
So they were islets that were treated the exact same way up until the perifusion. They were cultured together. They were handpicked, cultured together overnight, and then we would pick the islets we would use for the perifusion, and the rest of them would get flash frozen for the proteomics. And all the proteomics was done in one batch, basically. So we collected 2 batches. We collected 140 donors. And then sent off the proteomics in 2 separate batches. Yeah.
Right. I just meant it's good to clarify, what are the amino acid content, the fatty acid content, and the glucose concentration of the islets that are going to go for proteomics. How long the islets sat in that mixture and what they saw before the proteomics happened.
Yeah, really good question. They were just in standard media that we use for human islet culture. It's just RPMI media supplemented with a little bit of FBS and pen strep. The actual concentrations of amino acids and fatty acids, I'm not too sure about, but we can just go back and look at that in the look up RPMI and figure out what's in there. We did also, I don't have time to discuss this today, but we also looked at, correlations with, for example, how long the islets spent in culture, for example, and to see if any of our, proteomics data set would change in response to, islets being in culture for a longer time and so on and so forth. So we have that analysis as well, if you're interested.
Great. Thanks for unpacking that, Jelena. It's, I know you guys had kind of stuck to the high level story a little bit because of the time constraint and the fact you're sharing this venue. Thanks.
Yes. Yeah, no, I'm happy to discuss as more as you like. But yes, I am just giving you a very sort of the highlights of the paper today. Yeah, and then the other thing we found, which was quite unusual, I'll go into that in a little bit, is we found quite a few proteins that were, the abundance of which was correlated with the fatty acid response, which is, to me, was quite surprising. Fatty acids are not thought to directly stimulate insulin secretion, or at least that's been shown in the majority of studies. Some studies have shown that certain combinations of fatty acids do affect insulin release. But sort of the main school of thought is that fatty acids on their own do not affect insulin secretion. But it turns out that we found, hundreds of potential regulators of insulin release in response to fatty acids, the majority of which haven't been looked at. So for example, PHB and PHB2 are prohibitin proteins. They're involved in fatty acid oxidation in the heart. But their role in the pancreatic beta cell, and particularly the human pancreatic beta cell, has really been largely underexplored. So again, these are pathways that we should follow up on and try and understand signaling, because fatty acid stimulated insulin secretion is really not well understood or agreed upon, really. So what about donors with type 2 diabetes? So of those 140 donors, we looked at donors without and donors with diabetes. 17 of our donors had been previously diagnosed with type 2 diabetes prior to their death. And so then we did some comparisons of looking at insulin secretion again between donors with and without diabetes. What you're seeing here, as opposed to those, hundreds of squiggly lines, is the average responses. So here we have the average response to glucose, leucine, and fatty acid in donors without and with diabetes. So here so G3 refers to low glucose, so 3 millimolar glucose. Then we have our stimulus, and then back down to 3, and then we have our second stimulus, back down to 3, and then finally KCL. And when we actually quantify this, you can see that donors with diabetes, of course, had a lower insulin secretory response to high glucose, which necessarily wasn't unexpected. But what was really interesting is that donors with diabetes did not have, at least in our hands, a lower response to amino acids stimulated insulin secretion. So when treated with leucine, 5 millimolar leucine for 40 minutes, we calculated the area under the curve, as you can see here. Leucine was able to stimulate insulin secretion in donors with diabetes just as well than in donors without, indicating to us that maybe these islets, are still relatively functional, just maybe their preference for fuel might be different, or their, signaling pathways involving glucose might be perturbed, but the ones involving amino acids might still be intact. So. And then, as you can see here, we identified, hundreds of proteins that were differentially abundant between donors with and without diabetes. Again, in red, we have proteins that are up in donors with diabetes, and in blue, proteins that are up in our normal glycemic donors. Again, we see things like phosphofructokinase, which has a known role in insulin release, but then we have hundreds of proteins that haven't really been explored in detail. And we don't know what they do in the beta cell. We don't know fully how they function. So we mapped a lot of these proteins out onto sort of a prototypical beta cell and to sort of see where they can be found within the cell. So a lot of them are mitochondrial proteins. Some of them can be found on the endoplasmic reticulum. A lot of them are on the insulin-containing granules themselves. So again, hundreds of proteins to follow up on and figure out function, signaling pathways, and how their pathways may be perturbed in a disease state like diabetes. So onto the website a little bit, the portal. So our data set has been incorporated into the Human Islet portal. And the really neat thing about the portal is that it's our paper has been published, but the portal will continuously get updated. And so more donors will continuously be added on this portal. And so since our paper, there's been 20 more perfusion profile data sets. So for 20 additional donors, and we need to update that again. We haven't sent our next batch of proteomics in, but when that is done, that will also be updated. And if you're interested in sort of learning about a little bit more about the portal and how it's used and how it was developed, there's a really good Sure Science webinar almost 2 months ago now by Dr. McDonald and Dr. Ewald, who gave a really good overview of the portal and how to use the data set, how to explore within those data sets, and then how to download data and so on. And I'll talk a little bit about that today as well. So I touched upon this briefly, but we talked about sort of this idea of going beyond glucose as a marker of, beta cell function. So most of us use glucose, and we use glucose at a relatively high concentration, I would say, to study or determine islet function, right? We're gonna use glucose anywhere from 15, 16, I've even seen 20 millimolar glucose to study how much insulin is released. And so To me, 20 is definitely on the higher end. Most of us should not be seeing 20 millimolar glucose within our bloodstream unless we are dealing with diabetes. So that's one thing we should probably discuss is whether, using different concentrations of glucose might be a better indicator of function. And then also moving beyond glucose, right? So again, glucose is the most well-known, recognized stimulator of insulin release, but we really should be moving beyond that and looking at other fuels, other nutrients, and so on. And so this question of whether fatty acids can stimulate insulin release, again, is still a little bit controversial. And if you remember from the slide I showed you earlier on, in mouse islets that did not have that variability, fatty acids did not stimulate insulin release at all on their own. And in human islets, though, the majority of the donors, also we did not see a very large secretory response, insulin secretory response in response to oleate and palmitate. However, we did see a very small percentage of donors that had this exaggerated response shown here in blue. And we're referring to these as fatty acid high responders. And honestly, the first few times I saw this, I was not convinced. It was a very unusual response. It's not something I've seen before, but I've also never studied this many donors with the exact same protocol, right? And so every 10, 12, 13 donors, we would see this really unusual response. And then we went back and, tried to look at the donor metadata, tried to look at the islet isolation parameters and try and understand whether, something here could explain to us why these donors had such a high hyper response to the fat when it comes to insulin secretion. And we couldn't find anything in the donor metadata or the islet isolation parameters. But when we looked at the proteome, we did see some potential regulators of fatty acid response. So we saw some proteins that were differentially abundant between donors that were classified as those high fatty acid responders and donors that were classified as low responders, or sort of, I would say, more typical what we would expect. And so if I am looking, or if you're looking, you have a data set and you're trying to determine, I have 50 or so proteins that I want to follow up on. Which proteins should I really prioritize? What should I start looking into more detail when it comes to my particular data set? And so I think this is where the web portal would come really, really handy, or at least it has for me. And again, Jessica explains this in a lot more detail, but we'll just go through it a little bit today. So there's three main views. There's the feature view, the donor view, and the omics view.
I'm sorry, this is Marcella. Are you sharing
hi.
your screen or are you just talking?
I have slides up.
Yeah, I can see them.
Okay, I don't see any slides. I wonder if you have two screens.
Oh.
No, I only have one screen
Okay.
at the moment.
That's very strange. Is there two tabs in the new Zoom? Look up top.
I see two tabs, that's right. So I didn't have it on the screen setting. There had been a bunch of updates for the Zoom, so my apologies for interrupting.
No, no
Okay,
worries.
no
But
worries.
great presentation.
No worries.
worries.
Thank you for letting us know.
Great discussion.
Yeah, please let me know if there's anything technical going on, because they will be switching screens in a second. So if you can't see it, please stop me and let me know. I can't see all the Zoom questions, so
I've got them.
okay, please do stop me. So yeah, so where was I? So I was talking about how you can use the website to sort of prioritize which proteins or genes or whatever you wanna follow up on within your data set. And so, for example, when it comes to these, fatty acid sensitive islets that we identified, there's sort of three main ways we can use the website to go through and try and figure out which list of features might be important for us to further follow up on. So you can go into feature view. So you can ask a question like, if you have a protein of interest, which metadata are associated with that protein of interest. You can go into omics view. So you can start looking at, proteins and pathways that are associated with a strong insulin and secretory response to fatty acids, or, what other question you may have. Or you can go into donor view and look at particular donors that you see had this exaggerated response to fatty acids, and then look at which metadata is associated with these donors. So I'm just going to be talking a little bit about, the feature view, and we'll go into the website in just a moment. So again, these are a list of proteins that are differentially abundant between donors that were classified as high responders versus low responders. So what I'm going to do now is I'm going to pick a protein, and for the purpose of the short talk today, decided on picking a protein that I actually didn't know much about, SAMD11. It's one of the more differentially abundant proteins between the low and the high responders. So it's up here, so it has a higher log fold change and a relatively high p-value as well. So I'm going to go into the website, and let me know if you can't see this. So I'm going to go into feature view here, and I'm just going to type in SAMD11. So we'll click on here, and now we're going to click on search. Okay, so I told you at the time, I did not know much about SAMD11. What is it? What does it do? So the website's really nice, where you have all these links to different databases. So you can click here on the NCBI database, and now you get information about what this protein is. So it's sterile alpha motif domain containing 11. It's predicted to enable several functions, including histone binding activity, protein domain specific binding activity, protein self-association, and so on. You can go down and see that it is found in the pancreas. It's expressed in the pancreas, albeit I would say at lower levels than some of the other organs looked at here, but it is in the pancreas. And then you can go down and look at some related PubMed articles, right, that are related to SAMD11. And so you can just have a brief search, brief look through this. It looks like there's only 20 citations in PubMed on SAMD11. And I had a look at these earlier, and not a single one of them mentioned beta cell function or islet function or fatty acid response, which is interesting. Okay.
So was it associated with exocrine function then?
So if we go back, the papers are on autism, and what else was there? Actin regulation, which is actually really interesting. But there isn't nothing to do with pancreas beta cell function that I could find. But it's just a quick way for you to, take a protein that you're interested in. Instead of Googling multiple things, you can go into the website, type it in, and follow up on these databases and see, quickly what it is, what it does, and some of the publications that are associated with it.
A little detective work.
Yeah, I found it really useful. And then you can go into this other database. This is the Common Metabolic Disease Knowledge Portal. Again, so SAMD11. So you can look at something called a huge score, which quantifies genetic support for the particular protein you're looking for in diseases, human diseases. And so, for example, what pops out to me here is type 1 diabetes. So maybe there's something to do with, beta cell function to do with this protein involvement. So that's another really neat data set database that's attached to the website. And then you can start following up on the donor metadata, the functional data, and so on and so forth. And you can organize it based off of, I like to do adjusted p-value, but you can organize it based on, highest coefficient, unadjusted p-value, or even omics type, if you just want to look at protein, or if you just want to look at gene expression and so on. So, for example, what we know about SAMD11 is that it's negatively correlated or associated with non-endocrine cell proportion. So if you click on this, you get a really nice graph. that shows you that as I don't know if I can break this up, but as non-endocrine cell proportion goes up within our donor preps, at least for the proteomics, SAMD11 abundance goes down. So it does seem to be associated with being found within the islets themselves. And then you can also look at, so this is now gene expression here. Here we're looking at beta cell proportion. So give it a second, as beta cell proportion within a donor prep goes up, the expression of SAMD11 goes up, telling to me, or at least indicating to me that it is found within the pancreatic beta cell. And then you can follow up on, for example, diabetes diagnosis. So donors with type 2 diabetes had lower expression levels of SAMD11, which to me, if I was going to follow up on this protein, is important to know. And then you can keep going and looking at, so for example, here we're looking at the insulin secretory response to olein palmitate. So as SAMD expression or abundance in this case goes up, the response to olein palmitate goes down, which is really interesting to know, and so on and so forth. So for example, you could take the list of proteins that I had that were differentially abundant between my two donor criteria, and then go through the website and kind of prioritize and determine whether you think it's worth following up on that protein to further explore function or signaling and so on and so forth. So this is one way I would be using the website to do that. So
It's a great resource to extend your hypotheses.
yes, I agree. Okay, just a few more slides before I let Howard take over. So yeah, so that's one way I would use the website. Just a few take-home messages. And again, I do apologize. It is a very, quick, high-level overview of the findings of our paper. And if you're interested in talking more about the paper or had any questions, please do feel free to contact me. I'm happy to talk about it. But what we did find, sort of the two main messages from the paper, is that, there's considerable heterogeneity of nutrient-specific insulin responses. So just like we saw, or we have known about postprandial blood glucose responses, the same seems to be true for insulin. And then also we really need to move beyond studying only glucose as a marker of function and start studying other fuels as well. So for example, like I said, with leucine, with the amino acid, we see that in donors with diabetes, leucine in particular is just as capable of stimulating insulin secretion in those donors as our non-diabetic donors. And then we really wouldn't have seen this, high fatty acid response in these subset of donors if we never tested fatty acids, right? If we only focused on glucose. So, and then finally, just like to thank everyone in the lab and all our collaborators and the funding agencies. And if you have any other questions, I'd be happy to take them. So thank you.
Great. Thank you very much. Yeah, in the interest of time, let's have Howard move into his slide presentation, and then you can both take questions at the end if that suits.
Sounds great. I will stop my share.
Okay. Can everyone see my screen?
Yep. Can you put it into, yeah, there you go. Perfect.
All right. Thanks again, Monica, for inviting us to present. And my name is Howard. I'm a postdoc in Jim Johnson lab. I was originally a wet lab biologist, but I became really into bioinformatics and help with different project and scientists. Which brought me to contributing to Jelena's paper. And as part of the paper, I'm going to present the comparative analysis of human islet transcriptomes and proteomes, and also showcase a little bit of the related functions in the human islet portal as well. So we all know that mRNA protein relationship is not linear because there are multiple layers of regulation that influence the abundance of mRNA and proteins. So our question is, how much do mRNA and proteins agree in islets? As Jelena already introduced, we got our islets and conducted RNA sequencing and proteomics. We also have some nanostring data for the transcriptome as well. So for the comparative analysis, we used the 90 common donors and over 7,000 common genes to compare the RNA-seq and proteomics data. And also to validate the RNA-seq data, we also compared it with nanostring data using the common donors and RNAs as well. And the raw data of this analysis can be accessed conveniently on the Human Islet portal by going to the data download tab. We can filter the dataset by data availability. And if we select both proteomics and bulk RNA-seq here, we can extract the 90 common donors for the two omics. And then for data download, we can select the different data sets we want. Here I have nanostring, proteomics, and RNA-seq data. And we can also download all the different donor and islet parameters. And from here, I did my analysis on two types of correlations we're interested in. The first one is called within gene correlation. It is for each genes, we compare the mRNA and proteins across all the samples. And this correlation will inform us whether when the RNA change, whether the protein or how well the proteins change with the RNA for each gene. And the other kind of correlation is a cross-gene correlation. So here we take the mean abundance of each gene for its RNA and proteins, and then calculate their correlation across all the different genes detected. So this correlation will tell us when an RNA is relatively higher compared to other genes, whether the protein is also higher in the cell. So before getting into the main analysis, we are wondering whether the RNAs measured in RNA-seq and nanostring agree, because RNA-seq could have some technical issues and noises. So the answer is yes. When we look at within gene correlation, we found that over 93% of the commonly detected genes have positive correlation between RNA-seq and nanostring. And for a cross-gene correlation, among the different genes, the nanostring signal and RNA-seq abundance also have a decent Pearson correlation R value. So, which means if the RNA-seq and protein later on don't have good correlation, it's not because the RNA-seq didn't measure the mRNA reliably. So now we can look at our first main question. Do RNA and proteins correlate within genes? Surprisingly, or maybe not that surprisingly, we only found around 19% of genes that have positive correlation between RNA and proteins, and 0.5% of the genes even have negative correlations. And when we look at the distribution of the correlation R value, it's centered at around 0.1, which means most of the genes don't have a significant correlation between RNA and the proteins, which is consistent with previous findings in other tissues. So what are the genes with good within correlations? When we do overrepresentation analysis using the positively correlated genes and using the gene ontology cellular component library, we found that the positively correlated genes enriched cellular locations like cell surface, vesicle, and other organelle lumens, and secretory granules, etc. And for the genes with non-significant correlation, for the genes without RNA protein correlation, they enriched protein transferase complexes, ribosome complexes, and some other organelle membrane and nuclear proteins. So next main question is, do RNAs and proteins correlate across genes? We found the correlation is quite moderate with the R value of 0.5, which is also consistent with other tissues. We also wonder, do RNAs and proteins correlate with the same islet parameters? And we found that for each islet isolation or function parameters, the number of proteins and RNAs correlated with those measurements are very different. Especially for the functional parameters, we found lots of proteins that have significant correlation, but not much of the mRNAs. But lots of the islet isolation or culture parameters correlate with lots of RNAs, which suggests that RNAs are might be more sensitive to those parameters as their changes are quicker than the proteins. We also want to ask, do differentially expressed RNAs and proteins agree? We found that the differentially expressed RNAs and proteins are from very different genes mostly. Only a few genes that are in orange color are differentially expressed in both RNA-seq and proteomics. But the correlation of their fold changes do have a moderate positive correlation. Our next main question is, do transcriptomic and proteomic changes enrich the same pathways? The answer is yes, they do enrich lots of similar pathways. And here we used all the detected RNAs and proteins, not just the common ones, because we want to use the entire detected transcriptome and proteome. And we used the Hallmark and KGG gene set libraries, and they enriched very similar pathways. And both transcriptome and proteome enriched many inflammation-related pathways, and they are upregulated in T2D, and oxidative phosphorylation and beta cell related gene sets are downregulated in T2D. The last question I'm going to present is, can transcriptome and proteomes give similar predictions of cell type compositions? So we can get cell type scores using a method called BISC and using known islet cell type markers and arsenic cell markers. And from the RNA-seq data, we oh, by the way, the cell type scores here are relative values within each cell type, so the scores across different cell types are not comparable. And from the transcriptomes, we found the alpha cell have higher scores in T2D, and beta cells and have lower score, and arsenic cells also have higher score. And similarly, from the proteomics, it suggests that the T2D have lower beta cell scores and also higher arsenic cell scores. Because we also have stem cell data from the proteomics, we found that the scores for stem cells are lower for all the cell types, suggesting that they have lower amount of all the cell types compared to primary human islets, which also makes sense. And related to this, I got curious about what kind of genes, proteins or RNAs are associated with beta cell proportion. And conveniently, the Human Islets portal have pre-calculated cell type proportions using a similar method as mine, but with slightly different twists. So under the omics view tab, I want to look at the linear regression between the protein expression and beta cell proportions. And I want to correct for the, I want to adjust for the diabetes status. So from here, I can get what kind of genes that are associated with beta cell proportion. And this is a graphic summary I can get from the portal. And when I click each dot, it gives me the detailed scatter plot. So this dot right here is glucagon, and it has nice negative correlation with beta cell proportion. And under the result table, I can also download all the genes with their statistics. And here it shows the top genes with significant association with beta cell proportion. And as Jelena showed, I use these links to read about these genes. And some of them are really interesting, but I won't elaborate here for the interest of time. The title is around here is proteins associated with beta cell proportions. But next, I also want to look at RNAs associated with beta cell proportions. So I did the same analysis, but just used RNA-seq instead of the protein expression here. And I can get a list of RNAs that are associated with beta cell proportion. And I'm sure you recognize lots of interesting genes that associate with beta cell. So that's my short presentation for today. And for a brief summary, we found that the islet functions correlates with more proteins than RNAs. And the individual RNAs often do not reflect the changes of the corresponding proteins in islet in terms of the fold change in differential expression or the correlation. But the overall transcriptomic and proteomic changes are both informative to identify the altered pathways. And also they are informative to predict the cell type compositions. And again, all the raw data can be conveniently accessed using the Human Islet portal, and the portal also have convenient functionalities to let us look into the genes and do some of our own analysis. With that, I'd like to thank the Johnson Lab, especially Jelena and Supervisor Jim. And this is a great team study with lots of different labs and scientists. With that, I'm happy to take questions, and thank you so much for your attention.
Great. Thank you both for guiding us and walking us through your use, basically your use cases of the Human Islet resource. It was great, and giving us a little taste of your newspaper and your work. So if anyone would like to ask a question, feel free to unmute yourself or drop something in the chat and I will ask it. This is a great jump off point for many people. Oh, Mark, you have a question?
Sure, I'll ask a question. Jelena and Hoa, thanks both. This is awesome. excellent resource, very clear presentation. Howard, I'm going to ask you a question on your conclusion that there is not all that much correlation between protein and message. And of course, that's an easy enough message because we typically consider, for most of the pathways that we look at, we consider proteins to be the doers of biology, right? So intuitively, it's an easy message to sell, but at the same time, it to a degree makes you wonder what we do with, like, I don't know, a decade and a half of transcriptome data. Do we throw it all out the window? I'm sure you're going to say no, and I agree with that. So I guess the question then is, how much of the moderate at best correlation between protein and mRNA is attributed to real differences in the biology of mRNA and protein? And how much of that do you think is rooted in the inevitable variance in both of those techniques? I mean, both proteomics and sequencings, there is noise associated with these data, right? So if you could deconvolute, which I know you can, but how much of that lack of correlation is simply rooted in the noise or our inability of measuring both proteins and mRNA even more precisely?
That's a great question. Thanks, Mark. So my impression is that when the data set is large enough, like for Jelena's data, it have close or over 100 samples. The comparison of both omics reflect the real biologies more than the technical noise, because when I do the same analysis with some of my other data sets that have low sample size, their correlation are worse, and also the correlation between the fold changes also became worse. So the impression is that with the large data sets, the consistency between the two omics are stronger, which are telling me that it's biology. Also, by comparing the NanoString and RNA-seq, their correlations are quite good. It's two completely different technologies with different principles. So that's also telling me the technical noise are not contributing critically to this discordance of the two omics. On the other hand, as I mentioned, the overall impression is that for the individual genes, the mRNA and proteins don't agree that well for most of the genes, although for a small proportion of the genes, they still have positive correlation. But so when we interpret individual genes, when we see a difference in the RNA-seq out there, which are much more available than proteomics data, we shouldn't interpret it as protein changes for sure. It doesn't mean the corresponding proteins are changed. But on the other hand, we show the overall transcriptomic changes and proteomic changes agree pretty well. So I think the overall transcriptome are still informative and useful. As you mentioned, there are lots of transcriptomic data out there. We shouldn't treat them as garbage, like all garbage. We can still take advantage of its power because transcriptome can have a higher coverage of genes than proteome. It might detect twice as much genes as proteome. So when we do pathway enrichment or other analysis that utilize the entire genome or transcriptome, I think it's still relatively powerful. And the pathways enriched are still reliable if we do the pathway enrichment correctly with the correct background and everything and correct. And if you understand the statistical method and use it correctly, I think it's still useful.
Howard, how standardized is the use of what you're talking about? Is the thorough use of this bioinformatics, approach that you're talking about that's needed? Do you think it's all over, or is it constantly done properly?
It is constantly evolving, but there are lots of like commonly or consensus method out there for sure. But it is a challenge that many people don't use the method correctly. Some people may not understand the methods thoroughly and just use any tools that's convenient to them. So
So
yeah,
that approach is evolving too, right? Seems like hopefully more people will get on board and use the correct formats.
Yeah, I want to mention a review paper I co-first authored called Hang on a Sec. Hang on a Sec. So the sec is SEQ. It's a it's a comparison of RNA-seq and modern microarray. The main take-home message is actually modern high-density microarray is more reliable than RNA-seq. But in that review and also some papers cited in that review, it discussed a little bit of how to correctly use the pathway enrichment, etc. And there are also some other good reviews discussing exactly what you're asking. So feel free to read those.
That sounds great. Great recommendation. Well, in the interest of time, and thank you everyone for such a great discussion. We will be editing this these talks and posting them online shortly. And thanks again for showcasing what a great resource the Human Islet project is, and hoping that others will dive into it and start to use it more robustly. Thanks again. Have a great rest of the day, everyone.